we are very excited to have Dr. Mark Davis and Dr. Vincent van Unen from Stanford University with us today to discuss their Crohn's research that PROPEL A CURE is proudly funding. Dr. Davis is a world-renowned immunologist and is the director of the Stanford Institute of Immunology, Transplantation, and Infection, a professor of microbiology and immunology, and a Howard Hughes Medical Institute investigator. He is well known for identifying many T cell receptor genes, illuminating how these cells recognize diverse antigens. His laboratory pioneered studies of the biochemistry, genetics, and cell biology of these molecules and T lymphocytes generally, which play a key role in orchestrating immune responses. Currently, he is focused on a systems level understanding of the human immune system. And interestingly, for Crohn's patients, his work has concluded that the variation in most immune system parameters are not driven by inherited variation, but rather by environmental factors. And Dr. Vincent van Unen, an award-winning postdoctoral researcher from the Netherlands and Rubicon Fellow at Stanford's School of Medicine, is working in the labs of both Dr. Mark Davis and Calvin Koo, using the most advanced technologies, including organoids, to unravel the mysteries of intestinal disease. His goal is to figure out what the environmental factors are, specifically antigens, putting a name to them that trigger IBD. So joining us today are a couple of our board members, Al Fliss and Angela Clark and Ildiko Mayes, as well as Lisa Kelly. And thank you all for joining us here. I'm really happy to see you all. So my first question is, can you share a little bit about organoids? And so what are organoids and why did you decide to pursue this path?
Vincent, you want to take that, or do you want to direct it at one person?
I'm happy to give my perspective on this. First of all, thank you for the kind introduction, Annabelle. It is a pleasure to be here. So my background in my PhD program in Leiden, I investigated a lot the immune cell composition in the intestine of patients with chronic inflammatory diseases, such as Crohn's disease and celiac disease. And based on that research, we identified that there were many specific immune subsets in the intestine to Crohn's disease that are different from celiac disease and that are different from a healthy intestine. So we are having an increasingly awareness of all the types of immune cells that are aberrant in the intestine of patients with Crohn's disease. Now, in order to understand how these immune cells function and through which mechanisms they may contribute to the inflammation these patients are experiencing, we need methods to basically investigate their functionality. And therefore, we were very interested in working with Calvin Koo and his organoid system, because organoids are basically a 3D culture system of growing mini organs. So from a biopsy of a patient with Crohn's disease in the lab, we can regrow mini organs of the patient in a dish. And we have been doing this already for 2 years successfully. And what is very interesting about these organoids in Calvin Koo's lab is that they do not only contain the epithelial layer of the intestine that is responsible for absorbing the nutrients. They developed as the first people, the first researchers, I should say, a way to make organoids that they also maintain the intestinal immune system. So the fact that we can grow a mini gut from a Crohn's disease patient that still contains the immune subsets that are likely responsible for the inflammation gives us an opportunity to study them in the lab, because then we can alter them, we can trigger them, we can remove them to really figure out what are the interactions of these immune cells that result into inflammation, in the hope to get a better mechanistic understanding of the disease.
Does anybody have any questions to ask Vincent specifically?
I just wanted to ask how many patients were involved in this cultivation or this initial stage with the organoids or the tissue that was produced.
Right. So we have 2 years of experiences growing them for celiac disease. And that is where we really established the model, especially postdoc Antonio Santos in Calvin Kuo's lab has really driven this research in setting up this organoid system. So all the characterization of it has already been well established in that setting. Therefore, we could quite easily implement it for IBD patients. And this is now ongoing work. And so far, we have grown organoids from tens, 40, 50 patients so far. And a large body of that research has been devoted in getting them to grow well, characterize them, at what time should we be looking at when, as the organoids grow, do they still reflect what's going on at the tissue level, and where is that window of opportunity? So there's a lot of characterization that we first needed to do. Now we have a good understanding of that. So now we are continuing in trying to figure out what is really going on with the disease.
Okay.
If I could just elaborate on that in my perspective. If for many decades, the dominant paradigm in human translational research was a mouse model of disease, and there are many mouse models of IBD, and yet they have not really produced anything of value with respect to human disease, and actually at least the clinicians I've talked to say they don't really model any of the IBD types, including Crohn's, very convincingly. And yet this was kind of like what everyone was focused on. And what happens is then these ideas take on a life of their own, and they become like the dominant model, whether they work or not. And about 15 years ago, I was, wondering why human immunology wasn't going anywhere. And in fact, people were abandoning it because it wasn't going anywhere. But they would just refocus on something else in mice. And I just thought that this is kind of crazy. And if we don't know enough about the human disease and the dominant model is not informing us, when is a model not a model? As Lewis Carroll might have said. And that is when it's not predictive, and yet again, these things sort of have a momentum of their own and funding, and they have leaders of the field promoting these things. And so I thought, well, this is not working, and we should be doing something else. And what we should be doing is looking directly at patients and analyzing patients with these actual disease, and not depending on these models, as convenient as they have been and are, because it's just in a mouse. We have to really get busy and figure out how to do this. And then you run into the thing, well, you can look at a lot of things. You can characterize diseases, and we're getting much better at characterizing diseases in patients. We've done a lot of that, and healthy people as well, because you don't know what the disease really is until you know what health is, and health has quite a broad range. But then you get to a situation, well, there are lots of things that you would like to do in patients, but that are illegal or immoral, and you can't do them. They would arrest you if you try to do this. So you needed some way to dig deep into the disease, and organoids have been the way to do that. Is if we can if we can mimic the disease, again, it's like, okay, we had this mouse model, forget it. Let's have an organoid model, because then if in fact is even better than a mouse in terms of being able to dissect the components. Because if you have it in a dish, it's just so much easier to work with and you can get so much more information.